Are we ready? Back? Yep. Okay. So this section is going to be on the GBS bioinformatic pipelines. It's an overview. So getting from your sequence files to genotypes. So I'm doing this presentation, but I didn't do any of the the coding of the pipeline itself. I wrote a lot of code to wrap it up and do the logging kind of things and the reproducibility and the stuff that I talked about this morning. And this talk today is going to be focused mainly on the reference-based discovery pipeline. And we'll tell you about the other things briefly, but this is primarily about that. So this is the philosophy of the GBS pipeline. So why didn't we just go out and use something somebody else had written, right? That's always a question. Yeah. Well, mainly for efficiency, and you're going to see where we get some efficiencies in this pipeline as we go through. But one of the big ones is we only align unique fragments once. So you can imagine that you could have see the same fragment 10 times, 100 times, 1,000 times, 10,000 times, depending on the size of your data set. And if every single time you encountered that fragment you had to align it, well, that's going to take up computational resources, right? So that was one thing. We wanted that. And then there are data structures that have been created that are specific to the data types that we're working with. Okay? So in some cases, this was kind of new types of data, and we wanted to be able to work with them appropriately. So I said that there were 3 pipelines. The one that I'm going to spend most of the time on today, and on which the hands-on tutorial is based, is the discovery pipeline. It requires a reference genome. There are multiple steps to get from the sequences to the genotypes, and like I said, the hands-on tutorial is based on that. There's a production pipeline. It leverages information from the discovery pipeline to call SNPs, or to actually just identify genotypes. It doesn't really call SNPs, does it? It looks them up based on information that we have previously found. And then there's the unique pipeline, which was covered. I really need to get rid of that, don't I? Because Fei Lu's, I really don't expect Fei Lu to be here tomorrow morning at 9:30. Not really. What's that? No, not if he's coming by Wellington, yes. Yes. So I'm not really going to cover that, but you've heard a bit about that already.
Katie just covered it.
Katie just covered it, exactly. Okay, so I want to talk about vocabulary here, and it's always kind of useful to speak the same language. Part of the reason I've asked some molecular biologists, some geneticists, and some bioinformatician or analysis to be in the same room is so. Part of the goal of this workshop is that we start developing some common vocabulary so we can talk to each other in a way that's meaningful about what we're trying to do here. So this is kind of basic GBS type vocabulary and beyond that. So sequence file is just a text file, okay? And it contains the DNA sequence and supplemental information. And in our case, we're using the Illumina platform, so it's the ones that's specific to the Illumina platform. A taxa is when we're using Tassel, this software device. We haven't even defined Tassel, really, have we? It's a software developed in the Buckler group to do lots and lots of things. The GBS pipeline is one subset of those things and does a lot of kind of genetic analyses. And we'll play with it tomorrow. But in that world, a taxa is an individual sample. So when you think sample, taxa, interchangeable in this context. So a key file is just another text file, and it's used to assign a GBS barcode to a taxa. And remember, this is part of the way this whole thing works is we use these identifying barcodes. And we had an interesting question in the recent workshop. They're not actually barcodes, right? They're just known strings of DNA. We don't actually get on make a little barcode on the DNA. It doesn't really work that way. And then a GBS tag is a DNA sequence which consists of a cut site remnant from where the enzyme cut, right? And additional sequence. And we'll see more about that in a minute. And then a GBS barcode is just, it's a short known sequence of DNA used to assign the GBS tag to its original taxa. Okay. This is just a schematic of the discovery pipeline. This one, I think, we've had many, many iterations of this diagram. This one is one that I think conveys the ideas pretty well. and is reasonably simple. So it has all these parts with the arrows telling you where things are going. You know, information is coming from the sequence and informing this, and so on and so forth. And finally, you know, the goal is to get from here, the sequence, to the genotypes, right? That's our goal. Okay. So we start with the sequence. And so these days you don't see these things, these QSeq files. This is an old file format, but it's really, really easy to take a look at and read, and so I like to use it to illustrate the point. We don't really need to pay much attention to this stuff on this side. What I want you to look at is over here, and we've got things a little bit color-coded. So APEK1, that enzyme, leaves a cut site remnant that's either it's CTGC or CAGC, because it has this wobble base in it, so you can get one or the other. And so you see in red, those are the restriction cut sites, right? Okay. And before those, there's some other letters. Those are the barcodes. So when you open up a sequence file and you look at it with its GBS data, right? Not if it's just random sequence data, but if it's GBS data, you should see some patterns kind of like this. Now, not everything shows the right pattern, so we've got some things that don't come through quite perfectly. And that's true with any system. So just to highlight that that happens. Okay, a key file. So remember we told you that we have 96 or 384 barcodes, but we've done 50,000 samples. Well, how does that work, right? And we also told you that we typically analyze all this stuff together. Well, how do you do that and not get everything confused? Well, there's a few pieces of information that taken together make it unique. So here's our barcode, right? Here's our sample name. And these are really, I mean, we put these here on purpose, but these are really poor sample names. You ever go to in the fridge in the lab and it just says 1? That's really, really bad. So don't name your samples like this, okay? Don't do that. Name them something meaningful. And we can get into details about naming things. Actually, Katie, do you want to do that now?
Yeah, I'll just say, because it's not in a slide anywhere, but a lot of times people submit things that are either technical or biological replicates. And if you think at some point down the road you want to merge that data in this particular spot where it says DNA sample, and when you're submitting, if you're submitting to IGD, when you're submitting your spreadsheets, make sure that those samples are named the same. Because we're gonna, with all the other information that's there, we're gonna know which one it was, so it can be separate if you want it to, but if you want it to come together at some point, you have to name the samples the same, otherwise it's a really big pain in the butt, okay? Because
that's putting it lightly.
There are ways to do this in the pipeline.
Yeah, so there are tools that are available to do this merging of things, and it can be done at the level of sample name, but there's also this column of library prep ID. So you know how we said before that sometimes you'll take a library and you'll run it multiple times, right? Okay, so if it's coming from the same tube, the same library, and it's the same sample, it will always have the same library prep ID. And at Cornell, we developed a system that as it goes through the process, it generates that unique identifier and keeps it in a database. And this is something as a community, as we start building up our own infrastructure, we'll have to sort out how best to do that together. Okay, so what makes a unique sample? What in here helps you identify the sequence and put it back? Well, okay, there's the barcode, that's one thing, right? But also this flow cell, this comes from Illumina. They have a, like a stamp on there that says this is the flow cell name. It's read by the machine and is incorporated into the file. So you've got a flow cell, and we know that there are 8 lanes on a flow cell. So within a lane, this is the lane. So this gives you a barcode within a lane, within a flow cell, and that will give you the sample. Does that make sense? That's how we can actually reuse barcodes. So we have to capture all of this information, right? So this is a really important thing. Typically, this information itself is stored in some kind of a database, and then when you actually want to run the pipeline, you generate this key file from a database, right? And how many of you would keep this in an Excel file? No! That was this morning's lesson. Don't do that.
Rob, is a relational database like Access any better? Or should they be done too?
Well, actually, we use a relational, we have used a relational database.
MySQL.
Yeah, MySQL. Yeah, and John's got a question and probably a lot of experience with this stuff. But I did ask you not. I did ask you not to answer me any questions.
I'll certainly with animals rather than plants, but you've got sample IDs, so you know we have problems with replicates as well. But the other thing is that we often have a lot of samples for a single individual. Plants that would be a bit difficult, but for a single individual, and so we would always have the individual ID. But I suppose everybody's going to now tell me that they're going to put 200 individuals into one sample ID or something stupid like that. So do you keep, how do you keep track of that? Because typically we have problems where the individuals we think the sample came from didn't actually come from that sample and vice versa.
Yeah.
We have DNA sample IDs as well.
Yeah, there's DNA sample IDs. You're not seeing it in the key file. There are additional data structures that hold this kind of information.
The library prep ID comes from a particular DNA sample on the well, in a particular library prep. So it's one of those wells on the plate where you would have stuck a certain barcode in on this one plate, right?
There's a lot of information that you might want to keep track of that may or may not currently be supported by your database. And I've run into this problem before, and so there are a lot of fields available in the database that the IGD is using, the HTS database, that I wasn't using, so I, fudged it and collected that information in a field that I wasn't already using. But if you're building your own system, you can build that into the system, right?
Yeah. And this is all around our talk about Kia and other things, and what do we do to generate this stuff. But it's really important that you do it and you store it right, and it can be double checked and so on.
Yeah, one definite usage of GBS is to find errors like that.
And when you're talking about merging, that's a really, really good point. I would merge things that are the same library prep ID. If those are wrong, if those are different, something has gone terribly, terribly
really
wrong at the level where you just want to throw out all your data anyway. I always checked if there were replicates on the plate. I always checked that they matched genetically, and I did that as another QC step, but also to see if those things could really be merged, because if it was not the same, it could have been not the same because somebody messed up when they're plating the DNA. But we also had issues where there was a vine growing in 2 different locations, and people thought they were the same, but certainly they were not. So there are multiple levels to this problem. So I always just checked everything that didn't have the same library prep ID genetically to see whether or not they were the same, and we identified some problems there.
Yeah, so Katie's just actually illustrated another use for GBS as a diagnostic, right? You can use this tool to check to see if what you have is what you think you have, and that's a good way to do it. Okay. So tags. This is a kind of a graphical representation of tags. So this is what we get. An illustration of a fragment from a GBS library. Not that we would sequence this whole thing, right? Because this insert could be huge, or 400 base pairs, and we'd never sequence it with the current length of Illumina technology. But we have things that we're looking for. So we're looking for good reads, and only the first 64 bases after the barcode are kept. And the reason for that is it's 2 longs in Java. In other words, it's a way of keeping the memory footprint of the software relatively low. This restriction is going to be lifted in, I think, TESL 5. And so this is another thing we haven't talked much about, but there are varying different versions of TESL. There's TESL 3, TESL 4, and TESL 5. There are a lot of things going on. It's a very actively developed program. We're going to be using 2 versions of TESL for our hands-on exercise. TESL 3 because for running the SNP calling pipeline, because it's pretty well stable, and Jeff has done a fantastic job of documenting it. And the other thing, and we're going to use TESL 4 for some of the visualization because it produces some reports that it has features that TESL 3 doesn't have. Okay, and you'll see how this works tomorrow. Okay, so yes.
That limit on the insert size, you say it's lifted from 64, so is it just going to be 128, or is it going to be 256, or is it
32 times X.
No, we're just thinking of having it anything you want.
Really?
Making it
But we
a
haven't-
user-defined, a user specified variable.
We haven't done it yet, but
parameter.
it's in our plans to
It's-
do
yeah,
that
in
for
it-
Tesla 5.
It's in the plans, yeah. So it's getting there.
No, we won't keep it as a string, we'll use whatever number of longs are required and just pad the end with with zeros basically. So,
Yeah.
but we'll keep track of how long, so.
And you'll see how that works in a second too. So so Goodread has the barcode, and does anybody know why we need the barcode? I'm looking for audience participation. If you guys don't have this one, we're really in trouble. Why? That's right. You assign it back to the sample. Okay. Okay. And then it needs to have the cut site because it's anchoring. We're anchoring all these reads to the genome based on the starting cut site, right? So we want that there, so we can make these comparisons and called SNPs, and then the rest of your insert DNA up to 64 bases. Okay. So that's what we're looking for. Sometimes we see it's a short fragment. So we've got the barcode and the cut site insert combination that are less than 64 bases. And then followed by another cut site. Okay, so that would be a short fragment, still useful. And then we also see sometimes these look exactly the same, a chimera or partial digestion.
Hit the
Oh, I
forward
think
arrow.
you have to go back one. maybe.
Oh, maybe not. I think I messed up these slides at about 4 this morning. I'm very sorry.
Okay.
What we should see with this is. What we should see here is a cut site and then the common adapter. Okay, that would mean it was a short one. When we're talking about a partial digestion or a chimera, you'd see the barcode, the cut site, and in a shorter insert, a cut site, another bit of insert. And this is a really, really important point to make that affects not just the bioinformatics, but it's generated from the molecular biology. And that's why I want you guys to kind of get a feel for both of these bits. When you do a digest, you're cutting the DNA up, right? Which is what we do. And if you remember from this morning, we do the digest. We cut the DNA up with a restriction enzyme. It has these sticky ends. It's in this well that has the adapters in them, right? And then we put some ligase in there. And the ligase just joins things back up, right? That's what it does. It's pretty simple. You cut it up and you put it back together again. But if what happens in the tube is that rather than getting adapters on both ends, you take a couple of these fragments and you stick them back together. You get this chimera, or maybe there's another, the partial digestion means it might not have cut, right? But you cannot tell the difference between these things bioinformatically. When you look at this sequence, you have no idea, right? So can you see how that would be a problem? If you had two pieces of DNA from different parts of the genome that came together, you sequence that, and then you try to align it to a genome and call SNPs, this doesn't make any sense whatsoever, does it? So that's a problem. So we can still use it, but what we would do is we would trim it at the end, and we say, okay, if we see another cut site followed by some more of your insert DNA, we're gonna just ignore the rest of that because we just can't be sure what it means. And then we have some rejected reads. This is the barcode followed by the cut site. Yes.
How often does that happen when you get one?
It's hard. It's
How often do we
You're
get-
gonna charge me money, aren't you? Because I'm gonna say it depends, and you don't know. So you can imagine. Let's just back up a second. It's a great question. I think somebody could probably determine it empirically if they had long enough sequence reads to read through these things all the way, right, and sort it out. But you can imagine that it would happen more often if you had too few adapters, because there would be more opportunity for your genomic DNA to ligate to itself. And it would be less likely to happen if you had enough or excess adapters.
What would be the adapter dimer, you mean?
No, this chimera.
Or the timer? Oh, I see.
And then and if you had excess, you'd also get the adapter dimer. Yes.
Right.
Well, hence the titration step, right?
Right.
Yeah.
Pardon?
You're only within 64 bases, and chances of having two pretty low. That's what I was
Oh,
Depends on your fragment
well,
sizes.
so generally it is, like actually having it happen, I think it is pretty low.
On
Do what you
about,
think.
in maize, about 20% of the tags that we get are less than 64 bases. So I think the expectation of the fragment sizes from AP1, I think, are 512 base pairs, but you get a big range of that by random. So you get a tail that are smaller, and we enrich for the smaller ones by the PCR. So,
Yeah.
so we see them pretty often in maize data. Something like 15, 20%, something like that, are shorter than 64. They're trimmed ones, which whether they're chimeras or not, doesn't matter.
So those are chimeras at the restriction enzyme step. But do you have chimeras at the PCR step, which will be more likely than the first one? Have you determined those?
Chimeras.
You can generate just PCR chimeras.
Like a PCR doesn't finish, and then it
Oh,
denatures and
I don't.
You have two templates annealing, and then, because you have, barcoded, BPA1, BPA2, and the other one, just amplify. And then I reckon in any case you would have more PCR chimeras than restriction enzyme chimeras.
I don't think I'm understanding what you mean by
well,
PCR.
imagine, okay, so you're doing in the middle of the PCR, you got these two prongs,
yeah.
right? You get something that doesn't finish,
Oh yeah.
and then it denatures, and then it lands on something else.
Yeah. So, but
And then
Yeah.
yeah, I guess that could happen as well. But would that be more likely to happen if it was a long fragment?
okay.
Is that
And it
would
might
that be
be
the logic?
also-
Would it be, would it if that's the case, I don't think it's
I still think you wouldn't see those necessarily as only the ones early in the cycles, like, they'd be mostly like these rare, if you clone PCR fragments and started,
yeah.
which is in a way is what we're doing here, but, so I'm sure that's gonna happen, I guess.
But to what extent, I don't know. And it does seem to me that it would be more likely to happen if it was a larger fragment.
And if the PCR goes too many cycles and things like
Yeah.
that. And
So
the
yeah, we
cycles
also
are too
we only
long
limit
and
this to 18 cycles.
are-
So,
Or the, yeah.
so yeah,
If it's a random event, it's going to be filtered out anyway.
yeah, and that's the other thing. It's gonna be, so yeah, so and a lot of times someone
That's
say,
true,
well,
yeah,
does
would
it
be
like
filtered
Katie
out.
says, a lot when she's talking about her, well, does it matter?
If it's
Can we
totally
use- is-
random,
yeah.
it'll generate some tag that's only seen once or something, and then
And it'll be
filtered out,
small.
yeah.
Yeah. So I don't think we certainly haven't measured that.
Well, sorry, can I just ask another question?
Yeah.
How short is too short? Because you're keeping ones that are less than 64, right?
Yep.
So how short is too short?
There's a
Depends on
point
species.
when they get really short, they become really repetitive, like it's, so then they're not really useful for calling
So
SNPs.
20?
Something around there, yeah.
So I was just thinking about what Katie said about the error thing, and if you're allowing one mismatch, 1 in 20 is actually 5%. So, if you're mapping to something that's you're doing the network filter, and
Right.
you're taking 20, you're allowing one mismatch for that topology, that's actually 5%. Which
John.
is way more than
Speaking, because we've sequenced the genome back when next generation sequencing was pretty crap, and we were looking at a lot around 20, 30, 40 reads, is if you have one mismatch in 20 or 30 base pairs, you won't actually be able to map it accurately onto the genome in the first place.
So so it would fail at the alignment step.
Right,
It'll
yeah.
fail at the alignment
Right,
step.
yeah.
Okay.
You really need for mammalian type genomes, that's diversity and everything else, you really need 50 or 60 bases minimum to do a good job with the alignments, minimum.
Okay. There you go.
I was going to say something else. That this is very similar to the design of the gamer size that you need for assembly. And it varies by species. So if you grab a couple of the available genomes and map them next generation sequencing data using different gamers, you'll see that the change from very, very short gamers mapping everywhere to suddenly becoming unique as you go with longer gamers. Actually, this change is very quick and it varies by species. So you can actually determine that pretty easily.
I guess what I'm asking is why do you just throw out anything that's why don't you not even keep anything that's not 64 as a minimum?
That's really the question.
Well, do you keep 63? 62?
I would
61?
not keep anything less than 60.
Then,
Well,
that's a
you could
filter
be
you
creating
can apply.
null alleles though if you do that.
Yeah?
Because you could have, at the same locus, you have some that have a cut site, some that don't. So some tags are 64, some are 60, or I've seen that happen. So that's one reason. But these are really good questions because
That's one of those fiddly jobs that you do.
Mm-hmm.
it kind of,
That has a lot of implications for what you say about.
yeah. because these are kind of like thought experiment things, and we do a lot of that, especially Ed, you know, when we're developing the whole thing, and when we're doing the bioinformatics, you know, we'll be presenting results, and he'll be going, that's not right, there's something wrong with your code. And he'll have done this little thought experiment with a little calculation and realize, you know, and the stuff we're talking about now is kind of, you know.
I guess you're right, actually, 'cause you want the coverage, don't you? So you don't care because of what you said about the missing data. You actually do want to remember that.
Yeah.
So, but definitely good discussion and good questions. But I think in the interest of me getting some sleep, I mean, we're going to carry on. So, okay, so we've talked about the sequence files. We've talked about the GBS tags, and then we've got this the tag counts. Okay, what are tag counts? You take the information from the key file, and each sequence file is processed, and the tags are identified and counted. Okay, and of course, we were just talking about the tags is shorter. If it's shorter than 64 base pairs, it's padded with A's, and information about the actual length is kept. And at this step, the tags and the counts are put into a one tag count file per sequence file. And here are a couple, either QSeq to tag counts plugin or the FastQ to tag counts plugin in Tassel 3 will do this job. Okay, and you'll see as we go through this presentation, I'll have plugin names at the bottom for most of the steps, so it kind of shows you what plugin is being used. And tomorrow we'll go through a hands-on exercise using this. So then there's the master tag counts. At this stage, we've got, you know, let's say 30 or 2 or whatever it is, tag count files. And we want to merge all of these together and make a master tag count file. And one of the things that we do at this step, this is really one of our first kind of filtering steps. And we specify a minimum count at the minimum number of times a tag has to be seen to be included in the master tag count file. So if you only see a tag one time, and you know this is another one of these fiddly knobs. It's going to depend on, you know, how many sequences are you putting in total, right? And what your research goals are. But if you only see a tag one time in a couple lanes of data, it's most likely going to be a sequencing error, right? Otherwise, you'd expect to see multiple times. So those get excluded. Okay. And then we've got this master tag count file. And remember I told you the philosophy of this pipeline is somewhat about efficiency in mapping the tags. So now we know what the unique tags are. They're all in this tag counts file. And we want to be able to use those tags and map them against a reference genome, but it's in the wrong format. The tag counts file format doesn't work with any aligner. So what we have to do here is convert them to a FastQ file. And the quality scores don't mean anything in this FastQ file. But that FastQ file can then be used with an aligner. So we take the tag counts file and we convert it to a FastQ file. And that's just a sequence file. So that's the tag counts to FastQ plugin. Okay. Okay, so we've done that, and we've got this FastQ file that represents the tags that we know about. And we want to take that and eventually make another file that we call the tags on physical map. So you align them to a genome, right? And then you put them in that format. So we use an external aligner, and most recently we've been using Bowtie 2 a lot. I think
We like BWA mem now.
you guys like BWA.
BWA mem is good, but
Okay,
I don't
so
think it's currently supported.
the point is that there are lots of different aligners, and do we have those slides yet? Those come up tomorrow. And yeah. So Jeff is going to talk about aligners a little bit tomorrow in comparisons of aligners. They don't all behave the same way. If they did, we'd have just one aligner, right? So, okay, but they all basically will end up putting something in this thing called a SAM format. That's what aligners typically output, SAM format. And then we've got a converter that converts the SAM format into this tags on physical map. And so then you have this file with the tags and where they're aligned. And there's I think we've got This is a text representation of the tags on physical map. It's I think it's a little bit scrunched, but there should be like a C or something in front of here. So there's the enzyme cut site. It's a little bit off the screen. This is sorted by alphabetically, which is why you see all these A's. Okay, just so you know, that's why that's there. It's not like real representation of the whole big file. But the things I want to point out here is, see, here's another cut site, the CAGC, and then it's followed by all these A's in red. So if you look in the length column here, so many of these are 64, but that one's 57, right? Because it's shorter and it's padded. So remember I told you we pad it, but we record that information. That's how it's done, okay? And then it also tells you how many times it mapped, which 0 or 1, which chromosome number it mapped to, which strand, the plus or the minus strand, and the start and end position. So that information is all captured in the tags on physical map file. Okay. So far what we've done is we've identified and counted the GBS tags. We've converted the tag counts to FASTQ, aligned them to a reference, and then converted the alignment to the TOPM. Is that reasonably clear what we've done so far? I mean, this is, okay. Anybody confused or lost? John?
Just double checking, at this stage you're only allowing one mismatch in those six, we'll call it 6.
Nope, thanks for bringing it up. That would be, so the mismatch thing, the one mismatch thing would be the non-reference pipeline. We're not even thinking about mismatches here. We're just aligning to the genome.
So whatever the aligner tolerates.
Yeah.
Yeah.
Yeah, and then you have to define that for your species. It's another dial, right? And it's useful to do some testing. This is why I spoke earlier today about testing things, and having external data sets that you can work with. Okay, so we've touched the sequence files once to do this tag count thing, right? And we need to go back through and look at them and say, well, which tags are from which samples? And that's what this tags by taxa file is going to contain. But remember when we created the master tag counts list, what did we do here? Does anybody remember what we did here when we created this? Come on, help me out. Louder. That's right, a minimum tag count. Thank you, Cecilia. Minimum tag count, right? So we're going to try to exclude some of the sequencing errors at that step. So we have information in here about things that are included, and then the stuff that's excluded is not there. So we use this file to inform what goes into this file, because there's no sense in putting things in here that we've thrown away at this step, right? Okay. Oops. Okay. So what we need to do this is the original sequence file, the key file, and I think that's pretty obvious now. Everybody understands what that does, right? Okay. The master tag count file, and those things together in this sequence.tvt HDF5 plugin will make a tags by taxa in this format. And I think this is the first time we've spoken about this format in this workshop. Is that right? Did we talk about this before? No. Okay, so this is HDF5 stands for Hierarchical Data Format version 5. This thing was developed in the fields of like astronomy and particle physics, and these people that have been making massive data sets for a long time, and they've long had a need to be able to store and retrieve information in really large files in some reasonably quick way. And so the Tassel coders leveraged this already available resource, and it's on version 5 now. It's not like it just was made last year. It's been used and used a lot. To start dealing with these really large genomic data sets. And when we talk about 50,000 maize samples by 100,000, like a million SNPs, could you imagine a table with 50,000 by a million? I think does that fit in Excel? So you need something a little bit more robust. So basically, this is for efficient storage of large data sets. It was first used in this stage, and you'll see a little bit later that it's going to be used for a lot more things as well. The tags by taxa, we do additional operations on this file. If you're running a large project and you're having, you know, let's say, well, I had 300 flow cells worth of data, right? 300 lanes of data, 300 data files. In order to get anything done in a reasonable time, you can chunk the jobs up across multiple machines and then create many, many tags by taxa files. Well, that's great, but you got to get them all together. So this modified TBT HDF5 plugin, one of the things it'll do is it'll merge a bunch of them together if you've created a bunch. This is a step at which you can merge taxa that were sequenced multiple times that Katie was discussing before. And it also, this plugin will pivot the TBT in preparation for SNP calling. So it's like the tables, in creating it, it's really efficient in this direction. But to use it for SNP calling, it's really efficient in this direction. So there's a plugin that'll just go, and that may take some time. But that's some of the things that the modified TBT HDF5 plugin does. Okay, so now we've got our tags by taxa, right? We know which tags are in which taxa. We know where the tags are aligned based on the TOPM, and then we want to call SNPs. Okay, so SNP calling. So you use those two files. And I was going to ask you again, Jeff, to talk about the key settings in the SNP caller. So the minimum F.
That's a minimum inbreeding coefficient. It's actually, if you're population genesis, it's an FIT. But it controls how much heterozygosity you want there. And if you have inbred lines, you expect them to have low amounts of heterozygosity.
Then the minimum minor allele frequency.
Yeah, that's pretty much self-explanatory.
And the
minor allele count is just similar to the minor allele frequency. It's just the number of actual observations instead of the frequency. They're very similar.
Yeah, but these two work together, right?
Those two work together, and you have to pass one or the other. So I often set the MN MAC, the minor allele count one, at a really high level that you can't attain, so that the minor allele frequency is controlling everything.
And then there's a minimum locus coverage.
And that's sort of how many individuals have been hit by at least one of the. So once you're calling SNPs, you're using the set of tags that align to the same start position. So I call that a tag locus. And locus coverage is how many individuals, how many taxa have been hit by at least one of those tags in that locus.
Yes.
Haploid.
Haploid.
Haploid, I think you'd want to have the minimum F up high, just like an inbred line. So you would never expect to see a heterozygote there. So you'd have min F at 0.8 or 0.9, or I wouldn't necessarily put it at 1, but yeah.
Okay. So the other thing that sometimes I forget to talk about is, so we've aligned all this stuff to a reference genome, right? And sometimes when people call SNPs, they take their alignments and they call against the reference genome. They say, is this different than the reference genome? But the way this works, there's a flag to get you information about the reference genome, but that's a separate thing. But what this is actually doing is it's making this locus, this tag locus, where all these tags align, and it's looking for SNPs between the tags, not between the tags and the reference.
Right, and but there is an option to include the reference. And I highly recommend that.
Yeah.
So there's a -ref option, and then you tell it the path to the reference genome, and it'll pull a tag out of the reference genome at the same spot, which is really good if you may not have a cut site in the reference genome. So you want to make sure it's there. In case there's an insertion or deletion, you can still tell where the SNPs are relative to that reference genome.
Okay. John?
There's obviously a little bit of magic in here too, because presumably you can have reads going this direction, reads going in this direction, and they can overlap.
Yes, but we define a tag locus as things on the same strand with the same start site. So
no, but you could have two tags that effectively pull the same stuff.
yeah, you do get overlapping, but we deal with that at a subsequent step.
At a subsequent stage, yeah. There's a thing called a merge duplicate SNPs plugin that
So
actually addresses that exact issue.
So you can get the same SNP read twice from different strands, from different tag loci.
That's exactly, yes, the point, yeah. that John's making. Yeah, absolutely. And there's a plugin that deals with that. So you call some SNPs, and you get out a hapmap file. And this isn't 50,000 by a million. This is just a small snippet of a hapmap file. So, okay. This is the general format of a hapmap file. When you get to this point here, where it says SGS, BRLL, and then it's got the colon and so on, that is the name of a sample. Okay, and I'm going to go and tell you what is in that name in a second. This is the site, so it's site1_2100. So this is kind of a coding for this, but you can see where it comes from. There's chromosome 1, position 2100. Okay, and then it has either A or G alleles. Now this table goes really far out, so you don't see necessarily all of this stuff here. But what you do see,
each of these columns is a taxon or sample's genotype, and there's only 2 names shown here, but there's actually as many names in the file as there are columns of genotypes.
But this is one of those deals when you're working with a text file and things get a little bit skewed. So the names here, you'll see, this would be the sample name, is SGS-BRL 067. That would be when you made your key file, that would be in the sample name column. And then there's a colon, and then there's something 633 Y5 AA XX. That's, I know, is a flow cell name, and it's a common way that Illumina names their flow cells. And actually, I was in on designing this business, so I know from that reason too. And then a colon and 2. Anybody want to guess what the 2 stands for? Lane. The lane. Spot on. And then there's a colon C9 in this case. Plate position. And that's used if there isn't a library prep ID. If there is a library prep ID, that shows up in the name too. And of course, you're holding on to all this information for diagnostics later, right? You might want to be able to easily go back and figure out where did this sample come from. Okay, does that make sense to everybody? Okay. So we've run the discovery pipeline, we've got genotypes, and there's many ways of filtering genotypes. Jeff's going to talk about that tomorrow, and I'm not going to go into it today. We'll do a real quick overview of the production pipeline. So why do we have another pipeline? So, well, when you do these big builds like these guys are doing in the Buckler lab, they can have many, many, many samples, and it can take months to do. Especially if you're trying to do software development at the same time as you're trying to do builds. We had a lot of fun with that, didn't we, Jeff? So, and the reason, by the way, the reason I was asking Jeff to explain the flags on the SNP caller is because Jeff coded that, just that SNP caller. So the most common alleles have been identified after the first few discovery builds. If you're getting a lot of, good diversity in there, you use this information from the discovery pipeline to call SNPs in a new one that runs quickly, and you can improve efficiency and automate some of this stuff. So there's a flag on the SNP caller that will update the tags on physical map file with the variants that are called, which essentially allows this to become a dictionary. Does that make sense? You say, oh, if I see this tag, I know what the SNP is. And so this is what we would call a variant modified tags on physical map file. And we have additional information over here. And this says, in position 28 of that tag, the SNP is a C. Okay, does that make sense? And so at the moment, the limit of that, or I don't know, I haven't been around long enough. Does that change? Do we still only have 8?
No, 16 is by default.
16 positions by default?
Yeah.
Okay.
So this is only showing one variant, but we can store up to 16 variants per 64 base.
Whoops. Okay, so my pocket. Okay. Okay. So you can also, when you get down to filtered genotypes, you can also use that information. So so you get these unfiltered genotypes, and then you do some filtering. You say these are the really robust ones, right? Like these are the ones that we test them. We do different kinds of filtering. We say these are the ones we really believe, and you can use information from the filtered genotypes to update this TOPM as well, right? So you end up with a TOPM with what is information about the most robust genotypes that you have, okay? And then You stick it over in the, this thing is then moved over into the production pipeline, and you go directly from FASTQ files to genotypes. So what it does is it looks up, it's a little more complicated than this, but it looks up the tag, and then no, that's pretty much it.
It's in the FASTQ
Yeah,
file,
just
read by read, processes the read down to a tag, looks for a match in the TOPM, and if
spits out
there's
a
a match,
genotype.
it says it adds to the depth of each allele
Yeah.
that's matched, and just runs through, figures out what taxa that's in, and calls genotypes.
So this is really fast.
So the idea of that is like we might apply, we do these discoveries periodically, but say you're doing genomic selection and you want to turn the cycles really quickly, the breeder doesn't want to wait until we do another discovery, he wants the answer right away. So in theory, we could, in the future we've already made our TOPM available on the website, so we could imagine breeders just getting it and getting their data back and just running it really quick, even through the GUI.
So did you hear what Jeff said? They've made a TOPM for maize, and they've made it available on the website. This is another way we can share information with one another.
So it's like a publicly available resource of all the SNPs that we think are good in maize, based on, it's actually based right now on 32,000 maize. We're up to 50,000 maize after we run production on the remaining stuff.
Yeah.
Yeah.
So does that, you see how we can work together and make things like really cool? Okay. There you go. So to run it, you need the sequence file, of course, a key file, obviously, and the production TOPM. And this stuff is still reasonably current. Tassel 3 standalone and raw reads to hapmap plugin. And
It's
when you run, it was still one lane processed at a time, and hapmap files by chromosome.
I think the hapmap files it's a little better in Tassel 4, but the documentation's not quite there yet. So.
Which is why we're talking about Tassel 3 today. And
most users right now will not really use the production pipeline. It's more people doing big scale stuff like us in Maine.
But we want you to understand that these kind of tools are available, and where things are. I mean, we certainly don't expect to use every single possible thing we've discussed today, but we want to give you the breadth of what we know about, so you can make good decisions. John, did you have a question? Yeah, sure.
I guess the question is also when do you put a border between discovery and production, and how often, or do you consider updating the TOPM from production, which means going back to discovery at some stage?
So, well, there's another one of those "it depends" answers, right? I mean, so if you've so let's just a simple example of this, and it's not, none of this stuff is trivial. But let's say you've worked out a pretty, something that a discovery build that you think has been pretty good. You've got good quality SNPs, and you're using it, for quite a while. But then you go out and you get some new germplasm that is substantially different than what went into that discovery build. You would probably want to do another one, right, and incorporate all that data into that new build. So that would be, one way of determining when you would want to do that. Does that make sense?
Yeah, because I guess the thing is that some people would actually be stuck into the discovery phase
forever.
Well, I don't know.
Yeah, could be, and it depends. Well, and it depends on. Well, again, it depends on, like Jeff saying, if it's genomic selection where you're crunching a lot of agricultural samples through.
I think as long as the founders of your breeding population are reasonably well represented, their diversity is reasonably well represented in the discoveries you've done so far, and you wouldn't need to run it again. But when you're bringing in new diversity that's not represented, you need to run it, a new discovery build.
So about that 20 minutes, is that per line? Or is
Yeah.
And what is that on what kind of CPU?
It was just a desktop machine.
That's still just a desktop?
Yeah, that was a desktop machine. Yeah. So I've talked about testing, right? And so we tested this production pipeline, right? We didn't just say, oh, here, go use it. But we compared hapmap files produced by the discovery pipeline and the production pipeline, and we did a site comparison. The discovery had 48,000, and production had 47,000, and this needs to be updated, because at the time, the TOPM could only hold 8 variants. And so the discrepancy between these two was that we had more variants found in the discovery pipeline than were able to be stored in the variant modified TOPM. And that was the issue there. It took us a little while to sort that out, but that's what, what it was in the end. And then there was a 99.98% correlation of the genetic distance matrices from either when you compared these two methods. So that's one way that we tested the production pipeline. Okay, and so just to give you, we've been talking about next steps in development, but the HDF format, we said it supports large data sets. They're going to put more stuff in there. They're going to start holding the TOPM, the TBT, the key files, pedigree file, other information in that repository, rather than in a lot of relational databases. There's always improvements to the SNP caller that are going on, and a major undertaking is the ability to use tags that are not present in the reference. You know, these are next steps. They're not here yet, but things that are actively being worked on.
So basically, there's no production pipeline for non-reference genomes, because if you don't have a reference sequence for your species, you don't have that TOPM.
In theory, you could do it because, as Katie's pointed out, that unique can now be a fork of the regular pipeline, and you get a TOPM, but the positions aren't real, that's all. But you could still store variants in there.
But the problem being is the positions are arbitrary. So if you run a discovery one time, you run a discovery a second time, those positions are not going to be the same.
But if you really wanted to do consistent calling across different things, then maybe that's a good way to go. Then, like we did it, we ran it in this head of most of our samples got this production, and then we wanted to apply more samples and get the same positions, and maybe that's a good way to do that.
I wouldn't suggest doing the production pipeline as a way to get around the fact that there are arbitrary positions between builds, unless you don't want to capture any new variation. Okay.
John, do you have another question?
Does that make sense?
Yeah, probably a bit over the top, but there's no way, you know, in your in your file, there's no way, you know, further downstream you might find that there's a whole lot that are parallel and stuff like that. There's no way to go back and edit that so that you get rid of those tags out.
Well, there was the sort of back arrow that Rob showed.
Yeah.
And
Oops, too far.
I think that's one place
This
where,
one.
even in TASL 3 documentation, I think we're a bit lacking there. We're missing that step. But yeah, we have some plugins, I think that Terry did, that you can use to, once you filtered out the genotypes, now you give it the list of the ones you want to keep. So some of those things you filtered out would be paralogs or whatnot. You've got other information.
Yeah, I know Terry wrote that plugin because I ran it.
Right, yeah.
So,
Yeah, I don't think I documented it though, so yeah. So I knew
so
about
this,
that.
but to answer your question, yeah. Okay. That is the end of that talk, but we have a little bit of, boy, we're way past 4:15. Overview of preparing for hands-on data analysis. This one is like super quick, if I remember right. Okay. We'll do this very quickly. Roger, you need to know how to do this. Okay. Okay, the purpose of this exercise that we're going to do tomorrow is to help familiarize you with the steps in running the reference-based discovery pipeline. Okay? And it's to show you a few Unix commands and generate a very basic SNP data set. Okay, that's the purpose. About the sample data set, first it comes from Maze. The sequence files and the sequence files contain sequences that are only from the first 20 megabases from chromosomes 9 and 10. This is a substantially reduced data set, okay? It runs quickly, that's why we're doing it. This is we've just beta tested this round at Deakin this last couple days, and it worked really much better than these longer things that we were doing that took forever and crashed computers and was just a nuisance. Okay, the key point and you're going to hear this from me from now through the rest of this workshop, that this is not meant to represent an actual analysis for your unique situation. It's not, okay? The flags and the settings that we're using here, don't think that those are appropriate for your stuff, because they're not, okay? And that's really, really crucial. So this is very good for testing of your software setup to make sure you got your computer set up and everything's working right before you actually launch into your own data, because it's quick, it's easy, and you get an answer. And it's also very good for testing changes in Tassel code, which is the original purpose of creating this data set. Okay. Okay. So you'll be working in a graphical desktop environment, a Linux environment, which is actually what I'm running up here today anyway. The commands will be run in a terminal window, so you'll be doing a little bit of typing. So we'll do filtering and data display in a graphical Tassel interface. That's be Tassel 4. The pipeline will be Tassel 3. There are some really basic commands. We're not taking you through, this is not like a tour de force of learning how to use Unix at all, okay? At all. It's a few things to help you get your mind wrapped around what this is and how it works. And the people that do bioinformatics is most of their job. This is going to be a breeze. For the people that do the molecular biology, it may be a little bit more challenging. But then again, the molecular biology that we've been discussing is probably a bit more challenging for the bioinformaticians than for the molecular biologists. So the whole thing here, guys, is you should be working together and helping each other understand by explaining the bits that you know best to your team members, right? Okay. So this is all on hand.